So most indications for our first infliximab biosimilar, known as either Inflectra or Remsima, the CTP13 molecule, were extrapolated based on this biosimilarity and by comparing to the reference product. So they were extrapolations, and the pivotal studies for the first biosimilar for infliximab were the Planatra and Planeta study in RA and in AS. And then based on that, it was extrapolated to numerous indications. As Steve had mentioned, the cost to conduct studies to achieve extrapolations, you wanted to test it in every indication. It kind of defeats the purpose of, in some ways, of the development of biosimilars. So in my opinion, I was convinced when I saw the initial data, the molecular data, and the analytics data. And if you build the molecule correctly, it can only sort of work the same, in my opinion. There could be minor differences, as Steve mentioned, which we'll talk about later with the protein folding and post-translational modifications, but it should be very, very similar as far as efficacy goes. So when I saw the initial data, that's when I was convinced that these things were going to be pretty much the same. So I'll take the next slide. Okay, so some of our early IBD data on biosimilars, because they were studied in other diseases first. This is a study that led by Peter Lakatos from Hungary, from Budapest. They looked at consecutive patients. These are patients who were switched to infliximab DYYB, the CTP13, the drug we really have the most data on is the CTP13. And so these were consecutive patients. It was open label, so no control group. The primary endpoint was clinical remission at week 14. There was no endoscopic endpoint, but they did have CRP data, which I'll talk about. And they only continued maintenance. As you see, the denominators are a little bit smaller there. And the folks who at least had achieved a clinical response by week 14. So in these patients, for the Crohn's population, the median disease duration was 6 years. It was 4 years for UC. The median CDAI score was 324 in the Crohn's patients. The median Mayo score in the UC patients was 9. Over half of patients at baseline were on corticosteroids, and over half were on azathioprine. And as you can see from the data, both week 14 and week 30, we have very good rates of response and remissions. Again, this is open label data, so that's why the rates are also so high. Obviously, and not surprisingly, higher in the TNF naive patients. This was not restricted to bio-naive patients. But also to mention, corticosteroid-free remission at week 30 in the Crohn's patients was 50%, and the UC patients was 56%. And the mean CRP had decreased by almost half in the patients, both with Crohn's and with UC by week 14. And then infusion reactions, less than 7% of the cases. And these were actually most commonly in the patients who have previously been on infliximab. And infections, 8%, but this is overall. As by comparison, a serious infection, pneumonia was only 0.5%. So in this study, they concluded that the biosimilar CTP13 is effective and safe and similar to infliximab as far as historical comparisons in both Crohn's and UC. So I'll take the next slide now.
Wait, Mark, can I ask you a quick question about that? I may have just missed that part, but for those who previously had anti-TNF, did they specify, were these primary non-responders? Did they develop drug antibodies?
So they didn't specify, but it seemed that there were mostly patients who were at least tolerating the drug and not doing terribly on it, but they didn't go into great details on who those patients were.
Thanks.
That's a good question. Okay, so now we have some randomized data. This was actually published in Lancet by Ye in 2019. It was a 16-country study in Europe and Israel, Korea, in the US, Mexico, and Brazil, so in the Americas as well. It was a non-inferiority study in Crohn's disease, 220 patients with moderate to severe Crohn's disease who were randomized to receive either CTP13 or reference or originator infliximab. Their non-inferiority margin was 20%, as you can see from the data that was more than generous. And actually, not presented here, but I'll talk about antibodies in a later slide. At week 30, the patients who were on these patients were then also re-randomized into either continuing their same drugs, so they're staying on infliximab or staying on CTP13, or switching to the other drug and then followed up until week 54.
Hey, Mark, can I ask a question right here?
Sure.
You brought up, and I think this is a good point because I want to get everyone's opinion on this. You mentioned for their study, the non-inferiority margin was 20% difference. You know, obviously in the other studies, Northwich was 15% difference.
It's 15% in most studies that I've seen.
Right. So who makes this designation?
Yeah, I mean, this was, to me, it was, I'm not sure why they selected such a wide margin. I mean, probably just because they could recruit less patients that way. They just wanted to get data. They already knew this stuff worked, so I think they were just trying to get it done. They didn't. I don't know to the details of how they decided that, but I imagine it was just for less patients done. But the data are so similar that if they had 15%, it would just been more patients. It would take longer, but it would have probably shown the same thing.
Wouldn't regulatory set the margin? I would think that the regulatory agencies would set the margin, right?
I mean, I think it was predominantly Korea and European patients. So I think, you know, the regulatory agencies in those countries maybe felt comfortable with already the concept because they were using it from other diseases. And so I think, yeah, that's sort of probably how it got decided. I think let's
Usually they use the exact same endpoints that were used for the originator, because that allows them to set the confidence intervals for them to achieve. And hence, you know, nowadays, so many of the indications that are, all of the indications used for the originator are usually used by the biosimilar. So you don't change it, for instance, a CDAI 70 to a CDAI 100 for the biosimilar. If Remicade was approved based on a 70-point improvement, those are the numbers that they're going to use for the biosimilar. And likewise, in most of these, like the infliximab ones, were done not in IBD, but in
RA.
In the RA. And the other thing is, all of these subsequent studies that have been done are really post-marketing.
Right.
They're really for us, not for the regulators.
And in this study, they chose a week 6 CDAI 70 as clinical response as their primary endpoint. And so, you know, they did block randomization. There's no endoscopic data, but they did have calprotectin. For these folks, the median disease duration was 4.8 years. Their median CDAI was just under 300. A third at baseline were on corticosteroids, and 3/4 had prior been on immunomodulators. And as you can see, they looked at also week 14, and the data were also similar. But as you can see, at week 6 and week 30, you know, incredibly similar rates of CDAI 70 and 100 response, clinical remission for both things. There were no for Calpro. They had similar mean between the two groups. Mean Calpros at week 6, 14, and 30, and the SAEs were similar, 5% versus 8%. So if you go to the next slide now. Okay, and so here you can see, I just want to focus on the right of the slide, the steroid-free remission. So this is corticosteroid-free remission at week 30. Was very similar, 46% versus a little bit over 50%. And antibody, this is overall antibody formation at week 30 was 39% versus 45%. And of note, neutralizing antibody was 20% with CTP13, 19% versus infliximab. So incredibly similar. And drug levels were also pretty much the same. So we can go to the next slide.
See, Mark has expanded the term from highly similar to incredibly similar. It's not a regulatory term.
Incredibly similar. Again, they built CTP13 correctly, so Celltrion did a good job. So now this is now we're going to talk about the idea of switching, in particular non-medical switching, which means that you switch the patient's medicine for reasons other than the patient's health and safety. So these are some data we have in IBD. So this is a Czech study from Prague, retrospective observational cohort. Wanted to evaluate the switch in both Crohn's and UC. It was a small study, 74 patients, 56 with Crohn's, 18 with UC that were on reference infliximab and switched to CTP13. The mean time on reference product was 3 plus or minus 2.2 years. About half were on concomitant azathioprine. At baseline, 72% were in clinical remission, 22% had mild to moderately active disease, and 5% had severe disease. Now, we typically think about only switching people if they're in remission or at least a very good response, but they sort of had a mixture here. Their outcomes, they looked at HBI for the Crohn's patients, SCCAI for the UC patients, the simple colitis index. And then they also actually ended up following them to week 56 as well. Their outcome is interest was disease activity and adverse events. So when comparing week 0 to either week 24 or week 56 for some of the outcomes, they saw no differences in CalPro or CRP at either week 24 or week 56. Infliximab levels were very similar at all these time points. For week 24 was 3.4 versus 3.8, as Rich should say, microgram per mil. Antibody formation was 9.5 versus 10% at week 24 and 9.5 versus 6% at week 56. Remission, as I mentioned, at start of study was 72%, and at week 24 was 78%, and also at week 56 was 78%. And there were no infusion reactions in the first 24 weeks. So they concluded that switching appeared to be safe and effective. Again, there's no control study, and the control group in the study is small, but there was some early evidence in IBD. There's obviously more evidence and larger evidence in other diseases, but there was some initial data here for IBD. So we can go to our next study unless you guys have questions. Okay, so now comes the Norswitch. So Norswitch was a Norwegian study of multiple indications. So it was not only IBD, but also RA, psoriasis, psoriatic arthritis, and spondyloarthritis. It was a randomized control trial, non-inferiority at 15% in patients who had been on stable infliximab for about 7 years, a mean 7 years, but had inactive disease for at least 6 months. And if you go to the next slide, just to show the study design. So you have 482 patients who were randomized 1-to-1 to either CTP13 or infliximab for 52 weeks. And that was their primary endpoint, was disease worsening at 52 weeks. And then they extended them for another 26 weeks, and they switched the infliximab patients to CTP13. So followed them to week 78. So now we can go back to the previous slide. For here, they looked at HBI and the UC activity index, which is essentially the Mayo score. And if you look at the bottom of the graph on the right, the primary outcome for all indications. So disease worsening was 26% with reference infliximab, about 30% with the CTP13. And in UC, they were very similar. However, in Crohn's, there was about a 15% difference in favor of the reference product, which initially caused a little bit of concern. And if you go to then the third slide, the OLE outcomes. Yeah, perfect. So this looks at the week 78 data. And here you can see that the numbers pretty much, numerically now, and in many of the cases actually favor switch, but not statistically, except for psoriatic arthritis, where it seemed to favor the reference product. But again, as collectively, there was really no difference. 17% for the group who had stayed on this their product, and then for the switchers at week 52, those who switched from infliximab to CTP13, 11.6%. So, the study was generated a lot of interest and a lot of controversy because it wasn't powered for each individual disease entity, and because of the small numbers, you're prone to getting kind of variable results. I know you've probably seen these data multiple times. When we saw the confidence intervals for the Crohn's 52-week data, it was definitely a little bit concerning. But again, it wasn't powered for just Crohn's disease. So you know, in Europe, they've been using it a lot. They've been using it for multiple indications. So I think that's probably why they sort of lumped them together. Also probably make, you know, recruitment easier. So I don't know if you guys each have your own thoughts about this study. Again, to me, if you make the molecule correctly, the clinical data will come out, you know, showing, you know, relative similarity. So we can keep going, or we can pause here for a sec, because the next section's on immunogenicity. So if you guys want, we can talk about some clinical stuff first, and we talk about immunogenicity, or I can just finish the section, whatever you guys want.
I think the important point in Northwich is a lot of the studies up to that had been in IBD, had been, you know, non-controlled, observational from all over the place. And this was the first randomized controlled trial in our disease state, showing with our disease state included, it was reassuring. So
Yeah.
To me, this is one of, if there's four important studies, this is one of them. So even though it's not powered correctly, I still think it's important.
Yeah. I wasn't concerned about the results because, you know, I saw the analytical data on the CTP13, so they made it right. So, you know, I wasn't concerned. You know, when you get into small numbers, you get into wide confidence intervals, and you get to some statistical chance, and then that's when you get sort of can get in problems. So, but with interpretation of data, but when you look at all the diseases together, the differences were very small, like 4 or 5%.
So Mark, if I were to, you know, kind of ask a question to you and us, as well as everybody else, you know, if we're presenting this data to a group of people who may not necessarily be as familiar with the biosimilar data as you, do you feel like this is convincing enough to tell them that they are very similar from a clinical perspective? We know on a molecular basis and as a construct, they're very, very similar, but do you think that these studies that you showed are enough? Or does everybody else?
I mean, I think the collective data we've shown, to me, is enough. You know, we could always put more studies in, but then like you gotta start losing your audience and get glazed eyes and stuff.
I think they are. I think it is as well. I just wanted to kind of get a sense. Sounds like it is.
Okay, so we can move on if you want to immunogenicity, and you can go to the next slide. So these are two different studies on the left and the right. The first is Geert de Hans's group, but it was a collaborative Belgian study from Netherlands. It was prospective, 120 consecutive IBD patients with Crohn's and UC actually, and their primary endpoint was an infliximab level at week 16. These were patients who were in stable remission for at least 30 weeks. They used the HBI in this case, and their primary endpoint was to see what the infliximab level at week 16 was and how that compared to week 0. And as you can see, at baseline, at the time of switch, the infliximab level was 3, mean of 3, and 16 weeks later, it was 3.25. Importantly, 86% of patients with Crohn's were still in remission. There was no significant differences in CRP or fecal calpro. Almost, you know, very few number of serious adverse events, and only one person developed antibodies. So this gives us our first sort of biological data. There was also data at Northwich that I didn't present showing for the open label extension, showing the drug levels being the same throughout the study for the two groups. So this gives a lot of reassurance that you're not going to lose response from a PK standpoint with the drug. And then on the right column, this is a study from the Netherlands, Frank Henshen's group. It's a single center, 2-year prospective study. They looked at 83 patients, so relatively small, 57 with Crohn's, 24 with UC, 2 with indeterminate disease, who were then switched after doing well to the biosimilar. They measured HBI and simple Crohn's colitis index, simple clinical colitis index. And if you can see, they looked at their primary endpoint was really drug survival over the 2 years. And the blue line is, the dotted blue line is discontinuation due to loss of response or AE. Some actually discontinued the drug just because they were in remission. So to me, that's the most important line to look at. The other line, which is not very, very different, is the overall drug survival. But you see over 80% of patients over 2 years stay on drug. their median infliximab levels at baseline week 52 and week 104 were 3.6, 3.7, 3.9. So this stayed really similar for stayed pretty consistent over a 2-year period. However, they were following some drug levels. So 14 patients had their dose increased based on either low drug level or losing response. And 5 actually had their level decreased due to either a higher drug level or being in remission. So only 2 patients in the 104 weeks developed antibodies, probably because they were following their drug levels. Another push for proactive TDM, guys. So we can go to the next study. So this is data from the Lancet study, the randomized controlled trial. And as I mentioned, at week 30, patients then who were on either infliximab or CTP13, half of them switched to the other product. So you had about 55 patients per group at week 54. And we saw the antibody data at week 30. So this is the overall antibody data at week 54. Now they didn't measure neutralizing antibodies overall for week 54, even though they had it at week 30 and they had new antibodies after week 30 and they measured neutralizing antibodies there. They just didn't add them together, which is too bad. But as you can see, they're pretty similar, a little bit numerically higher for the folks who are on infliximab reference, who are switched then to CTP13. But new antibodies after week 30, importantly, these were all low titer and only a very small percentage were neutralizing. So gives you some nice confidence that immunogenicity will be the same for the 2 drugs. And also, conversely, if you're developing antibodies, you should use the biosimilar, as Steve had already mentioned. So we can go to the next slide. So this is a study also from led by Peter Lakatos from Hungary, a prospective study in IBD, both Crohn's and UC. They had 136 Crohn's patients, 38 UC patients, prospective 174 patients. They're all switched, in this case, from CTP13 to infliximab. So this is the first data we're seeing switching from the biosimilar to the reference product. And if we start on the right, before that, the clinical remission was measured by CDAI or partial Mayo score, and they were similar, about 80% range at 8 weeks before the switch, at the time of switch, at week 16, and at week 24. And if you go to the right part of the graph, this is overall data. And this is broken down by drug level being either super therapeutic, greater than 7, adequate in the range being 3 to 7, and subtherapeutic, less than 3 micrograms per milliliter. And their mean infliximab levels were 5.33 versus 5.69. So after switching, so at week 16 after switching, that was their primary endpoint, 16 weeks after. And when broken down by disease, you see very similar proportions of patients in all of those in those 3 ranges, both for Crohn's and for UC. And antibodies baseline versus week 16, also very similar, 16.2 versus 16.9%. They didn't measure neutralizing antibodies, but again, gives us some PK confidence that these things are very similar and that we can monitor them the same way. So now we can move to the next slide. So this sort of defines, and you could make an argument to have this study, this slide also in the first section that Steve did, sort of defines how we think about transition, switching, interchangeability. So a single transition going from the reference to the biosimilar, or going from the biosimilar to the reference. A substitution study where you have a single switch and you can go back and forth, each drug switch to the other drug, or is interchangeable where we have 3 or more switches, multiple switches. There are 2 studies in psoriasis, one with etanercept, one with adalimumab. The etanercept study had three switches. The adalimumab study had 4 switches, showing similar PASI scores and antibody formation in the 2 groups. And they follow them, primarily to week either 30 or 35, and then extension period to week 52 or 51. So, and then Ray had mentioned there was an ACG abstract, and Miguel said there may have been something from Europe. So we could potentially even include those in this data set showing interchangeability in IBD. Miguel, wanted to ask for the European stuff, were they switching between multiple products like infliximab reference and then 2 different biosimilars going back and forth, or was it just one biosimilar?
It was back and forth to the same.
It was, but at the UEGW, I thought it was actually three or 4 infliximab biosimilars, if that's what you're asking.
Yeah, going back and forth between them.
That's my understanding. It was, I don't know if anybody else was in that. I think it was, Of course, I don't remember the exact presentation, but it was UEGW. It just was last month.
Okay. Okay. But that would give us some confidence in it.
Yeah, it's obviously not published yet, so it's just going to be abstract form.
Yeah, because with interchangeability, for me, the issue was if Euclid's transitive property applies. So if B is similar to A and C is similar to A, are B and C necessarily similar to each other, right? So, and when you talk about minor differences in tertiary, quaternary protein folding and post-translational modification and glycosylation, if that stuff over time going from one product to another, back to another one, if that is going to end up mattering, and also not all infusion reactions are antibody mediated. So that's going to play a role both in immunogenicity and in infusion reactions, that we still don't know. But studies like that, especially if they have a little bit longer follow-up, would be great to help answer that.
Yeah, I'm going to try to, while we're on now, I'll try to pull it up and see if I can find it.
Thanks, Miguel. We can move to the next slide. So this is data from Shamron Ben-Horn. He's one of the world's leading TDM experts and has done a lot of work in this field. And he did some of the early work on biosimilars as well. So there's a paper published and got 125 patients from Israel. And this study shows nice, very, very similar cross-reactivity of antibodies. The antibodies to infliximab similarly recognize CTP13. In this case, they use the product Remsima, which is pretty much the same as Inflectra. So they had 2 different Remicade batches and 2 different Remsima batches. And what they found was that all anti-Remicade ATI negative controls were also negative for anti-Remsima ATI, and all positive anti-Remicade sera cross-reactive with Remsima. And not only the negatives were all negative and the positives were all positive, but the titers were very similar and the correlations were almost identical. So this gives you a lot of confidence that immunogenicity is going to be sort of conserved between the products, and that it suggests that these products, these reference and biosimilars, have similar immunodominant epitopes. And that was actually also shown. It was a multi-country study published in APT in 2018. This author was Gonsalves, but it was with Shamron, Geert de Haans, Peter Lakatosh, and Silvio Danesic, sort of combining their data, and they identified 13 regions of infliximab, which were sort of the immunodominant epitopes. And there were 6 variable heavy regions, 3 constant heavy regions, and 4 variable light regions. So this also, they sort of furthered the data that were implied by this study by Shamron and showed that there really were immunodominant epitopes, in which this also gives you allays some fears in terms of switching between products and multiple switches, that potentially from an immunogenicity standpoint, you might end up being okay. So we can go to the next slide. So this is sort of a little bit of a summary slide. What does the real world evidence say about using biosimilars in IBD? So the FDA allows for extrapolation of cross indications, as Steve mentioned, and available data to date suggests that biosimilars for the anti-TNF products behave similar to the reference products. The drug assays for reference products are expected to work similarly for the biosimilars, as we mentioned. Anti-drug antibodies to reference products will cross-react to biosimilars and probably very, very, almost identically, as Shamron showed in the other data about the immunodominant epitopes being the same. So, providers then, I think, can feel pretty comfortable starting a new patient on a biologic, and also, to me, switching them, especially if they're stably in remission. And potentially even if they're not, we don't have much data on that, but it really should be okay. As far as concerns, we already touched on that. No biosimilar in the U.S. has interchangeable designation, although we're getting more data. We have some data in psoriasis. We're getting more data in IBD. We do have some concerns about the double and triple switches for the reasons that I mentioned. There are no published data on mucosal healing efficacy, although, again, to me, the analytical data is what was most convincing about these products. As there's a question of whether we should use concomitant immunomodulators when double or triple switching. And, from an immunodominant epitope sort of standpoint, probably not. But, you never know if there's minor things that end up mattering occasionally for patients. And again,
But we can all, I'm concerned about these last two statements, having rereading them again, because I think they're just generating concerns that are not, that you can always ask questions about this stuff.
Yeah.
But, the whole point of our discussion is you would treat it the same way you would treat the originator. And I don't, it's kind of asking us to look under rocks where these are really concerns from the originator companies that have welled up. I'm not sure we should actually propone those concerns.
Yeah, we can axe those. I'm fine with that.
Hey, what do others think? I mean, Ray, Miguel, Tina, you guys, do we propone those concerns or not?
I mean, I really don't think it's different from the originator biologics. So specifically for biosimilars, I agree with you, Steve, that it's not very unique to biosimilars per se.
I mean, likewise, this no published data on mucosal healing. You don't have data on other drugs that we have without mucosal. I think it's focusing on issues more from the originator companies that are, kind of fake news.
Yeah.
Generate.
I think the third point is silly about the mucosal healing. I mean,
the first two seem like they're the most important ones.
To me, I agree.
We can get rid of the other ones. That's fine.
I think the biggest concern is the double or triple switching because we just don't have the data on it.
Yeah,
but I do agree. I think the last three are sort of raising questions that probably aren't necessary because the role of concomitant immunomodulators is the same for reference as it is for biosimilar. But I think that it's almost better to have the considerations be a little bit longer than the concerns column. But just from a perspective standpoint.
Eyeball test.
I mean, the only thing I would add is, honestly, the concerns to date are minimal. I mean, I think even the word concerns is not a concerns. These are just additional considerations.
I mean, we could say unknowns or I guess the person's not really an unknown. Yeah, we could just. I think we could just leave it as considerations, and the first. Not have
or further questions or something to that extent.
Yeah. I just agree with Steve that looking at that, I think the whole point of this is to reassure providers about biosimilars and likewise so that they can educate their patients, because this is happening in the real world. People are already getting double and triple switches. It's happening as they move from one home infusion to the other. So it's already here. So I agree that somehow that framing there is negative. So I think we have to fix that.
I think if we change it to limited data or further studies are needed for double and triple switches, because it could be safe, we just don't know, right? There's limited data.
Yeah, we could just have limited data for that one thing. That'll be fine.
We do have data. I mean, the psoriasis study with adalimumab is a multiple switch study that could give them interchangeable status if they wanted, and the PASI scores are completely superimposed. So
yeah,
I mean, there's no reason to think that anything else would perform differently. So
or we could leave it as a fifth consideration and just be like, there are two good long-term studies in psoriasis showing no concern with multiple switches with between one product and another, just between two products, not between multiple products. But they're limited data in IBD, or maybe they're not as much limited data in IBD after the ACG and UEG.
Rather than using like mucosal healing or concomitant immunomodulators, I mean, most of the switches are non-medical switches. So I think that one of the concerns is switching during active disease, for example. And there is no data for that. Most of these are non-medical switches. So even the double switches, but yeah, I mean, I think all of these could just be considerations as one table and just keep it simple.
Okay, we can go to the next slide. We're almost done. So all of our prior studies were comparisons to infliximab. So this is for adalimumab, and this is our very own Steve Hanauer. So this is the Voltaire CD study. So looking at safety and efficacy of adalimumab ADBM, the BI product, compared with the European approved reference adalimumab. So patients were randomized to either reference or ADBM, and then they were switched at week 24. The ones who were on reference were then switched to ADBM, and they're followed up up to week 56. And the primary endpoint was CDA response at week 4. And if you just show the next slide. So here we go. This is a 70-point CDA 70 week 4, 90% for the BI product, 94% versus for reference adalimumab. Very similar, and safety is also very similar between two treatment arms. No unexpected safety signals. So I mean, you'd expect this to work the same as the if the agencies approved it as a biosimilar, meaning all analytical data are correct, would expect this to behave just like the infliximab biosimilar data. So no surprises there. And that's the end of my section.
I guess one of the questions is Miguel or Ray brought up, there were a couple of other presentations at UEGW, which I'm not familiar with, but ACG, which I am familiar with, that are, again, they don't say anything different, but they expand the IBD database. One was looking at the national, I think Nabil Khan looked at a national database, and Rush, I think also looked at, no, Rush was looking at different things. But there were several that we might, if we feel that this section that Mark just presented needs more oomph or modification. Miguel?
Well, I just found, actually I was wrong. I think it was DDW, and I found as part of the best of cornerstones, I can't remember who else might have been on that, but there are a couple interesting biosimilars. There was the efficacy and safety of the sub-Q CTP13 to IV. I don't know if that's interesting or not, but the one that I was remembering is the switching from.
I would caution Miguel about that one simply because it's really for a new product. Which
I know.
Injectable.
So but the one that I was thinking, there were two biosimilar switch ones, kind of one that's more similar to Norwich, but the other one was actually switching infliximab originator to CTP13 to CB2. And it's small, but it actually did not show any differences. So this is kind of a multi-switch or double switch, and they compared double switch versus single switch and didn't find a difference. And that was Mazza, M-A-Z-Z-A, S is the first initial, and it was presented at DDW. I can send the kind of the cornerstone PowerPoint that has the abstract for that, and then the other single switch one, which is not going to be too different than Norwich.
And just thinking back, there was also, I didn't present this, but there was that huge study from the annals, like 25, it was like 5,100 patients. 25
I just sent it to you, Mark.
Okay.
Annals of Internal Medicine.
It was 2,500 patients per arm. It was just real world, and the graphs I remember were like pretty much superimposable. It wasn't randomized, but it was enormous.
It's the largest study to date. And then I put in a chat box the link for the VA ACG poster, which also has some CTP13s going to SB2, I think, in that study. So that's in the chat box, the link there.
Okay.
It would be helpful to include at least one of those because it is happening in real life. And I think that there needs to be some reassurance, especially since a lot of these switches are not necessarily within provider or patient control. So, and it does have people antsy about it, and having at least some preliminary data, knowing that future directions are certainly needed, but future studies are needed. I think it would be reassuring for this particular context.
Yeah, the annals paper, that was just patients who were started on either. But there wasn't a switch study.
But it's better than the one study you showed.
Yeah, we could put that one in. That's fine.
And I think there's nothing wrong with showing the multiple switch psoriasis data, even though it's a different disease.
Yeah, I mean,
I don't think
I
it's
have those
reassuring.
slides. Yeah, I have slides on this the two psoriasis studies, the Egality and the Humira one.
It'll make us look smart if we talk about PASI scores. So that's another bonus as well.
Yeah, I can send those slides. Because those were long studies. There were a lot of patients. They were done to 52 weeks. They had a bunch of switches, 3 to 4 switches. Like, to me, that's pretty convincing data.